Hi, this is Dr. Dan Guerra coming to you from Authentic Biochemistry Studios in the inland Pacific Northwest. Today is, of course, the 18th of May, 2020. And as I've advertised on my Facebook page, I want to give you a lecture on how steroids impact the immune system. They're intimately involved, both the endogenous regulatory steroidogenesis, steroids binding to the receptor, alteration of gene expression pathways, that I've been going over in the audio Authentic Biochemistry lectures, but also more intimately and more discreetly and sometimes much more powerfully when we use pharmaceutical forms of steroids to modify the immune system, basically to suppress the immune system, and how those drugs have been tremendously useful in controlling autoimmune diseases, which can become quite dangerous, high morbidity, and sometimes mortality. Certainly a rather poor existence when you have a very powerful autoimmune disease like lupus, all the way to where these pharmaceutical steroids or steroid knockoffs can be quite dangerous because they suppress the immune system so well that you can get spontaneous infections. And more than that, disrupt the normal homeostasis of steroidogenesis, all the way back to the level of cholesterologenesis, which is, of course, the precursor pathway for all of this. So let's get started with this right now. Okay. So, again, this is an Authentic Biochemistry podcast video. And what I want to do today, the reason I'm doing a video is because I want to show you pictorially some of the ways that steroid mechanisms control both the facilitation of distribution of certain steroids, basically because we're focused on it, the immune response, and how feedback mechanisms control the synthesis and degradation of those steroids. And again, how exogenous application, that is pharmaceutical biomedical application of steroids, bifurcates this pathway, sometimes allowing you to have all the downstream effects, but not having any of the upstream effects, that is the regulation over the control of the expression of the steroid pathway. Understanding that it's not happening by itself, it's not happening in solitude, that steroidogenesis is happening with all the other metabolic regulation that is purposely coincidental with it in terms of homeostasis and vis-a-vis the immune system, because that's what we're discussing today. All right. So this is a hypothetical model depicting what I'm calling potentiating effects, potentiating events and effects, I guess, to expose what used to be called the paradoxical effect of insulin on protein phosphorylation. Now, this isn't a discussion of insulin, it's a discussion of steroids. It's a good example. It's a good way to proof out some misconceptions in biochemistry. Particularly what's misconceived is that you think that you turn on a pathway with something like, for example, insulin is synthesized by the pancreas when there is high circulating glucose, or also known as the well-fed state. So the well-fed state means that high circulating glucose needs to be taken up from the blood and introduced into cells. So the level of glucose is maintained homeostatically. And we know that diabetes is when you have hyperglycemia, you have too much glucose in the blood. So insulin is the rheostat that controls that. And it's regulated by the level of circulating glucose that can be measured by the beta cell of the pancreas, along with fatty acids and insulin itself, and also levels of other hormones that are circulating. So the process of insulin secretion, synthesis secretion, is very complicated. But that's not the whole story. Once insulin binds to its receptor, the downstream processing of that signal is where things really matter. So insulin by itself, it doesn't do anything, right? It's a peptide. So it has to bind to its receptor, and its receptor happens to be an autophosphorylating tyrosine kinase type receptor. So a TK or else TKR. And TK, tyrosine kinase receptors, or receptor tyrosine kinases, sometimes they're called RTKs in the oncogenic literature, basically do this. They have, this is a very simplified version of it, but you have an alpha subunit, which is extracytoplasmic, and you have two beta subunits. So you form a dimer of each of these heterologous subunits. Each one has a tyrosine residue. These tyrosine residues, because they have a free hydroxyl group, can be phosphorylated, make a phosphate ester of that tyrosine oxygen atom. Now, when that happens, this very simplistically will show you that one pathway can trigger a secondary messenger, such as the inositol phosphate cascade, IP3 cascade, that we've talked about at great length over the years. I keep saying that about so many pathways, but I know I have. And we'll get back to it, don't worry. But right now, just understand that there is an inositol cascade pathway. And so what that triggers is, right after this pathway is turned on, a phosphatase mechanism. So phosphatases remove phosphate from proteins. And particularly, this is a protein serine threonine phosphatase. So any proteins that are recognized as substrates by this phosphatase, which is triggered, that have phosphate on a serine or threonine, that phosphate will be removed. That's what it means by protein dephosphorylation. Now, when that happens, depending on the particular pathway you're looking at, will generate catabolic systems. Okay? So you're breaking down certain biomolecules and generating what? Sometimes generating energy. Okay? Now, that seems counterintuitive, that you could have catabolism at the same time. Now look at the left-hand part of this graph, or this slide, anabolism down there at the bottom where I made that red block. So here, the same protein, this is the tyrosine kinase receptor for insulin, the insulin receptor, or the IR, as it's usually called in the literature. Once it binds to ligand, insulin, triggers this response of autophosphorylation, and then that phosphotyrosine can go on to phosphorylate a kinase enzyme. So a kinase enzyme is an enzyme that itself is autophosphorylated often, so it is a kinase to itself, and then it is itself able to catalyze a reaction where it's phosphate from ATP. The gamma phosphorylated ATP is added to yet another protein. So it's called a kinase, right? So what you're phosphorylating are those same proteins, those protein serine threonine kinases, that on the other side, you were dephosphorylating, you see? So when you phosphorylate these kinases, they go ahead and further phosphorylate or act as kinases to the final substrate protein. Now, that can induce anabolism, okay? Such as the building of, getting ready to make DNA replication go on, getting ready for cell division, right? Now, that in biological sense is perfectly okay. It's not really a paradox, it's a pseudo-paradox, because if you want to get ready for cell division or cell growth, you have to make more macromolecules. You're going to go through cell division, you're going to finish that part of the cell cycle and do division. You have to be able to replicate DNA. That takes a lot of anabolism. You have to, first of all, make sure nucleotide biosynthesis is up to snuff. Then you have to take all those deoxyribonucleotides and replicate the genome before you ever make the business of making cell division, right? At the same time that's happening, you have to make enough lipid so that membrane can be ready for that cell division. So the cholesterol genesis has got to be jacked up, fatty acid synthesis has to be jacked up, phospholipid sphingolipid synthesis also has to be increased. So you need catabolism for energy to fuel all that energy consuming process of anabolism, but you also need anabolic pathways. So this can actually occur in the same set of cells. That's why these kinase systems are not just in one dimension. They're in multiple dimensions. They're all four dimensions. They're all spatial dimensions that are occurring because these proteins that are being phosphorylated are moving around the cell. But beyond that, you also have a temporal dimension. So what could be happening anabolically could be slow and steady and exist until another cell division, whereas the catabolic pathway could be turned on early to make enough ATP to fuel that system. So there are temporal differentiations there. So because there's a temporal differentiation, this does not violate the major rule of logic. Now, why would we even bring up logic in biology? Because biology is very logical, right? The reason it's logical is because logic is what reason tells us is not contradictory when it indeed is a sound event, right? And so you can't have direct contradictions in the cell, and that would mean that you would have a middle term that would always be involved there. That is, you would have catabolism and there and is an actinism copula, anabolism at the same time, right? You can have catabolism and anabolism in the same cell, but at different times. You get the point. So these changes in these cascades, there's rhythms that have to do with the speed of the particular pathway that's being finally translated through the cell, okay? That's one way that you do not have a paradox at all. You know, remember what a paradox is. It's like you get a result that makes no sense, yet that seems to be the result, right? It doesn't make any sense. It's an innocuous bit of evidence. And a lot of people say, well, that's paradoxical. Never paradoxical, because the cell is able to do this. Now, beyond what I'm showing you here, understand that when you stimulate a pathway, if it's insulin or if it's a steroid, stimulating the expression, let's say, of anti-inflammatory cytokines, and then suppressing at the same time pro-inflammatory cytokines at the transcriptional level, at the level of chromatin remodeling in the nucleus, upon stimulation of, say, a corticosteroid, that you can't have that system always on. You turn on the light switch and you can't leave it on. It's not like when you need the light on all the time until the sun comes up again. The light switch has to be able to be rapidly turned on and off. Or that is, the anabolic or catabolic grade has to be leveled much more rapidly in a cell. Because if you don't do that, if you can't compensate for a triggering mechanism, then you end up continuing to fire. You continue to fire, you overshoot your mark, using that particular metaphor. So you always have to have the brakes being ready to be applied at the same time you're accelerating. And that is all spatially and temporally. And all of the possible Baroque nature you can think of the biochemical pathways. So there are no paradoxes there. In fact, even when you have pathology, it's not a paradox. The pathology is also a very biological pattern, right? It's something that happens because homeostasis has been corrupted, such as in a very, very major disease in the world today, and that is obesity. Probably the worst health crisis in the world today. Not probably, certainly, is obesity, because it is the confounder of all other underlying diseases, at least contributing to it, sometimes directly correlated or even arguably causative to things like cardiovascular disease and cancer, autoimmune diseases, metabolic diseases. You get the point. Okay, let's move on. So let's get this a little bit smaller here. Okay, now this is from a nice diagram I lifted from the Immunoassay Handbook, 4th edition, 2013. It's an open access handbook. And then, now we've gone through this much better than this slide shows what's going on in the central nervous system. All they're showing you is higher brain centers. You're having cortisol affecting the hypothalamus, and hypothalamus is affecting the anterior pituitary, and cortisol can also work via feedback mechanisms on both the pituitary and the hypothalamus, right? So the anterior pituitary is responsible for making the adrenocorticotropic hormone, ACTH, and then that's going to generate in the bloodstream, ultimately downstream, cortisol, right? That's going to work through the adrenal cortex. That's where you're going to end up getting the final synthesis of the cortisol. So the adrenal cortex is responsible from, this is the HPA axis, hypothalamus pituitary adrenal axis. And finally, they're going to make cortisol, and cortisol is going to be in the bloodstream. Now, what does cortisol do, right? What does it stimulate? This is the solid bar. This is the solid arrow. It's going to increase glucose formation. It's going to increase fatty acid metabolism and mobilization out of the adipose tissue and out of storage triacylglycerol droplets in say skeletal muscle or even in the liver to some extent. It's also going to be involved directly in proteolysis, and it's going to generate an anti-inflammation paradigm. It's going to suppress inflammation. This is why synthetic corticosteroids are used in medicine, to suppress the immune response. Now, if you're a layperson, you always hear about being immune compromised, right? Immune compromised sounds like you don't have enough of it, right? You're deficient. That's not at all the valence you should be considering. Often the immune system is very, very powerful, very potent. And because the immune system is going about the business of destroying cells that are diseased or damaged, or that are synthesizing proteins at really high concentrations, therefore corrupting homeostasis in those cells, in that tissue, and maybe throughout the body. The immune system is there to fire off and destroy any of the cellular machinations that are not appropriate for normal health, growth, and maintenance, right? So this is the idea that people often miss. An immune response is a very powerful response, but it has to be tightly controlled. And so when you use synthetic corticosteroids in medicine, you're suppressing the immune response because the immune response can be very deadly to cells, tissues, organs, and indeed the human themselves. The entire life can be taken away if the immune response gets completely hyperimmune, hyperactive. This is what corticosteroids are used for. So it's not often that when people present in an emergency room that they have very low immune response. They may have a hyperimmune response, and they're immediately put on corticosteroids to prevent what corticosteroids are to prevent what the immune system is doing, which is increasing your temperature, increasing your blood pressure, increasing your heart rate, and increasing the killing of cells. And sometimes, for example, natural killer T cells or any of the innate immune responses can sometimes trigger via pro-inflammation, via cytokines and chemokines, the destruction of cells that are only peripheral or adjacent to those which are, say, infected with a bacterium or infected with a virus. We went over this way back in January when I was talking about this coronavirus that came from China, that an overshooting of the mark of an immune response to it is not what's probably indicated here if you look at the SARS and MERS data. It's more that you can have a precondition of an inflammatory response, and one of the ways that occurs is with any diseases I spoke about just a few minutes ago. Cardiovascular disease and cancer and metabolic disease. You have a steady status of a hyperimmune response. And that can all be enveloped under, again, that global health crisis, which is obesity. Gives you a hyperimmune response. So that's the large picture, hopefully condensed into just, I hope that was only a few minutes. So this is, again, from that same immunoassay handbook to remind you of what we're talking about here. So higher brain function, the central nervous system, any stressors, such as being exposed to very cold or frigid conditions or very hot conditions like in the desert or on top of a mountain in February. Any injury that can include any kind of contusions or lacerations or any kind of bruising or damage to the body. Of course, infection, which we've been talking a lot about, bacterial, fungal, viral, parasitic. Hypoglycemia, right? When you have very low levels of glucose in circulation, you can obviously go into coma if it gets really bad, but before that you can just simply suffer from syncope, that is fainting. Excessive exercise, very loud noise or loud noise in general, such as a screaming child or such as a rock concert, right? Or any loud crash or bash, or like a volcano erupting. Anesthesia, okay? If you're under some kind of anesthesia, either because you're in surgery or because you've taken a drug that is an anesthesiatic, the same kind of process can occur. And pheromones, this is it from insect population, but also there are pheromones or pheromone-like compounds in the human system that are related to biological and behavioral activities. Pain and any kind of toxic insult. The one I always like to use is ethanol. Ethanol is a neurotoxin. Ethanol, that is ethyl alcohol. That is alcohol that you have in your wine and your beer and your spirits. All of that ethanol is a neurotoxin. Okay, I like to say that every chance I get. Anyway, the higher brain then working through the hypothalamus and then synthesizing the CRH and AVP, right? We've talked about this before. Remember, these are the initial phases of the HPA axis, the hypothalamo-pituitary-adrenocortical axis, right? So there's the hypothalamus, there's the pituitary, there's the adrenal cortex. We've gone through this many times. CRH is corticotropin-releasing hormone. The AVP is the arginine vasopressin. We already know most of these other ones. TSH, if you don't remember, is a thyroid-stimulating hormone, also known as thyrotropin. PRL is prolactin. The alpha MSH is the alpha melanocyte-stimulating hormone from the pro-opiomelanocortin system. LH is luteinizing hormone, VIP. Maybe we haven't talked about that very much. Is the vasoactive intestinal peptide. And CGRP is a calcitonin gene-related protein. Okay, so that's enough of looking at the acronyms. So you trigger response hypothalamus, you make these releasing hormones. They work, again, through the pituitary to synthesize ACTH. ACTH goes on to trigger in the adrenal cortex the production of corticosterone or cortisol. And so then you generate this cortisol. Now what happens? It's in the blood. It's binding to receptors, something I'm going to get to in a minute. So it goes to peripheral tissues. It generates alteration of glucose metabolism, protein catabolism. We just went through this, right? At the same time, there's a negative feedback loop, right? So all of these glucocorticoids inhibit the CRH, the ACTH, the TSH, the PRL, the LH, and both testosterone, and they also modulate insulin way up at the beginning of this arc, way up at the hypothalamus. So you can see what can trigger at the pituitary. Oxytocin, estrogen are positive. Substance P, acetylcholine, which is, of course, a hormone. And antipeptin, which is another hormone. Those are all negative on the pituitary axis. PRL, progesterone, the alpha MSH, gonadotropins, all of those are positive on the adrenal cortex. And the modulators of the adrenal function include cytokines, VIP, CGRP, opioids, synthetic and endogenous, neurotensin, substance P, and neuropeptide Y. I left this one out because the literature doesn't follow that very much. There's been a lot of conflicting discussion of the adrenomedullin pathway, so I didn't bring that one up. But all of those then are going to be involved in maintaining this HPA axis. Okay? So that's how you control this pathway. Now consider that when you're taking exogenous pharmaceutical grade corticosteroids, you're down here at this level. So you don't get the regular feedback control because you're not controlling the entire axis, okay? Which has to do with releasing hormones and then the second stage in the pituitary and then finally at the adrenal cortex. There isn't enough feedback regulation here because these synthetic corticosteroids don't have the same bindings capacity as the authentic corticosteroids that are made de novo in vivo in the human body. So this is a little bit about what goes on when the receptor binds. Calcium channels are opened up. Here's the hormone binding to its receptor. It could be linked to a G protein. You can have adenylate cyclase firing cyclic A and P. Cyclic A and P dependent protein kinases like protein kinase A functioning to phosphorylate a series of enzymes. and other proteins associated with the mitochondrial activity. You can have a lipoprotein receptor, lipoprotein binding, vesicular transport of cholesterol. So this is showing that cholesterol is imported. Why is there cholesterol in lipoproteins? You often hear in the popular press, oh, LDL cholesterol is carrying all this cholesterol, and therefore cholesterol carried by LDL, the low density lipoprotein, is somehow a dangerous event. That's how you get cholesterol to the periphery, because not all cells can synthesize enough cholesterol, particularly secretory organs, necessary to generate all the glucocorticoids to be able to have enough biosynthetic pathway at the level of cholesterologenesis. So they rely upon, and it's necessary for lipoproteins to deliver the cholesterol to those given cell types, like the adrenal cortex, so that you make enough corticosteroids. So this is important to understand. If you lower constantly, chronically, using drugs that are always low, like HMG-CoA reductase inhibitors, which are given to people with so-called hypercholesterolemia to control things like cardiovascular disease, you also can be suppressing the generation of corticosteroids upon stress, which is an immediate response. And therefore, you're uncoupling that regulation. Understand that, please. So anyways, all of these components, all these protein kinase cascades, the influx of calcium through the ER, there's a phosphatidylinositol cascade pathway working through various other hormones. This is all happening simultaneously, that you're getting a stress response. And ultimately, the mitochondria, the biostatic pathway in the mitochondria, is what is used to synthesize the steroid hormone. Right? And then it's sent into the cytoplasm, and they can diffuse ultimately or bind to a protein. Right? So you need free cholesterol, you need cholesterol transport, and all these numbers here and pathways are showing you where there's potential corruptions in the pathway. But you need to get cholesterol in the mitochondria, you need to get side chain cleavage enzyme, that's the SCC enzyme, that's mitochondrial. That's going to take cholesterol, remove some of the side chain on that backbone, and then get that pre-steroid complex ready to go through any number of pathways to correctly make just the corticosteroid or not some other steroid hormone. Right? Of course. So that's the overview of steroidogenesis. So here's ACTH binding to its receptor, working through a G protein, making cyclic AMP, activating protein kinase A. Here's the lipoprotein carrying the cholesterol through. Right? We just saw, so this is like a more definitive type slide. This is the action of ACTH, remember that's the adrenocorticotropic hormone, on adrenal fasciculata cells to enhance the production and secretion of cortisol ultimately. So this is like a fine-tuning of what you just saw. So you need the lipoprotein to bring in cholesterol. They arrive as cholesterol esters, they're stored as cholesterol esters. So you need the cholesterol esterase, the enzyme that removes the fatty acid. That fatty acid can go on to do something else, which we've talked about, such as the production of other lipids. What other lipids? Phosphoglycerolipids and sphingolipids. Okay, of course. Sorry, I shouldn't have put that too far out of the way. Ultimately, you get this side chain cleavage so that you take cholesterol and turn it into cortisol. There's pregnenolone in the pathway. It's all happening in the mitochondrium. So cortisol is secreted. Now, the reason it's in quotation marks is it can diffuse because it is a lipid, right? And once it gets into the blood, of course, it binds to anything that has. So where's the regulation? Say, well, it can just diffuse. If it doesn't facilitate diffusion, some of you good cell biologists in graduate school or something say, well, wait a minute, well, how can it just diffuse? Wouldn't that be losing control? Well, yeah, except the fact that you have receptor-mediated events downstream. So only those cells that have the glucocorticoid receptor, the GR it's called, affectionately, only those cells will be stimulated by the circulating cortisol. Okay? So that's enough of control, and then therefore regulating at the upfront, right, upstairs in the vestibule at the opening of this whole process, the amount of cholesterol that's used for steroidogenesis. Control that pathway, and we know all about that control because we know all the upstream pathways leading from the hypothalamus to the pituitary, with all those releasing hormones and activating hormones. Ultimately, when you get to the point of synthesis of cholesterol in the adrenal cortex, you've already gone through 2 or 3 levels of regulation. So the level of cortisol itself isn't as important as all those regulatory components which will give you feedback inhibition. Right? And that's why taking exogenous glucocorticoids that are given in say an emergency room, that completely circumvents that pathway of regulation. And that's where there can be danger for the suppression of the immune response after the initial gain you get, because the immune response perhaps is out of control at the point where the person's entering the emergency room. It's a paper published in ACTA Obstetrics Gynecology from Scandinavia, just published April 30th, so a couple weeks old of this year. See, this was actually a EPUB ahead of print. And what does it say? Antenatal, that means before natal, corticosteroid therapy in COVID-19 pathophysiological considerations. Okay, this is a brand new paper dealing with this virus. So let's take a look at what it says. It's caught my attention. Okay. Severe acute respiratory syndrome coronavirus, the Chinese pandemic, can be associated with obstetrics. So that is something to consider. People are pregnant during this so-called pandemic. So clinical recommendations from the AOGS for this infection includes administration of corticosteroids as an adjuvant therapy. Okay? We know this because it's a hyperimmune response from having this infection, right? Or coincident with already having a hyperimmune response because the underlying morbidity diseases such as cardiovascular disease or the drugs you take for them. Right? Okay. So corticosteroids may diminish the inflammatory response, a major factor for lung damage and acute respiratory distress syndrome, which is the underlying danger in this particular disease. However, in previous SARS and MERS outbreaks, there were reported delayed viral clearance and no survival benefit, and actually an increase in disease severity upon glucocorticoid administration. Okay, so this is already known. It's in the literature for almost 2 decades now. It appears that this current virus, like SARS and the SARS interferon response, which I've gone over previously, and I'll show you again, displays a sequence of biphasic progression. Remember, interferon is going to be a very important cytokine in controlling toll-like receptor-mediated activation of the innate immune response, controlling things like infection. So there's a biphasic progression where an initial mild presentation of, say, a viral infection is subsequently followed by respiratory failure, characterized by a massive production of pro-inflammatory cytokines, right? See, hyperimmune response, and chemokines, which is causing cellular trafficking, even in the coincidental decline of viral titer, the well-described, what I call hyperimmune response. Question then arises, question that these authors had and that I've had. Early or late corticosteroid therapy, when you get someone entering the ER or entering a hospital or somebody with a viral infection, when do you administer corticosteroids to suppress the immune response, right? Because the immune response is what's giving you the morbidity and ultimately, in rare cases, the mortality. Because there are no toxins being generated by this virus. The virus isn't coming in with poisons and destroying cellular metabolism. This is all to do with your immune response, right? That's why glucocorticoids are given early on. So, given previous evidence on interferon responses in the SARS outbreak several years ago, a later corticosteroid therapy would clearly be indicated as more beneficial. That is, after that biphasic, hitting that second phase, okay? Because you want interferon to do its work. And interferon will be shut down because the glucocorticoids are going to inhibit the synthesis of interferon and the interferon receptors, the IFNARs, and all the other pro-inflammatory systems, which are going to control the titer of the virus. So there's two things happening. You want to decrease the viral load, that is the amount of viral replication, but at the same time allow for the immune system to stop that, prevent that, diminish that by the induction of the immune system, which has been triggered by the invasion/infection. Glucocorticoids are going to come in, especially pro-administrated or administered at the same time this replication cycle of the virus isn't fully taking off, and then you miss the mark, right? You miss the rhythm, you miss the sine wave, and then you can get a suppression of the immune response and then an explosion of viral replication. And furthermore, downstream processing, which can ultimately lead to increased amount of immune response. Okay. However, clinical emergency practice typically involves, this is what I've heard and read in the literature, the current literature, immediate massive anti-inflammatory therapy. And this could be associated with more disease severity and maybe even morbidity and maybe even death. Okay? So the administration of glucocorticoids has to be very well timed in these kinds of situations. So here's a paper, Phases of Inflammation, Stress Axes, way back in 2012. Okay? Now again, what I tell you when I give you these authentic biochemistry lectures is I'm telling you that I go into the scientific literature that's already gone through peer review, and examine what's already published, and then look at, of course, what's being published currently, and try to develop an architectonic, a large, complete understanding of the system that involves evidence already procured by carefully controlled experimental design, the execution of those experiments, the production of data, the frank investigation of the data to determine what is evidence from the data, and then a verification of the evidence to suggest a given new idea that then becomes another hypothesis in the scientific method. So you can see here that we're talking about initial anti-inflammatory and pro-inflammatory systems way at the initial phases of a response. So there's initiation, there's resolution, and there's termination. And we're going to be looking at the levels of norepinephrine, very high at the beginning as compared to cortisol. Norepinephrine still high at the end of the initiation cycle compared to cortisol. Then during the resolution time of this mediation of an inflammatory response by these local hormones, like this leukotriene B4, this is a leukotriene made from the lipoxygenase pathway, which is an oxygenation of preformed polyunsaturated fatty acids, of the omega-6 positional isomers, usually a very long-chain fatty acids. Omega-3 fatty acids can also be oxygenated, but because they all bind to different receptors and because they are substrates for those reactions, they often act not necessarily contradictory, but in a way that is coherently and I guess conveniently distributed against the process of the omega-6 oxygenation. That goes way back to lipid metabolism, something I talked about a lot because I'm a lipid biochemist. But remember that this initial phase is innate immune response, local hormones, leukotriene production, generated from something like arachidonic acid that's been released via phospholipase A2 from a preformed phosphoglycerolipid in a membrane in association with a stressor like an infection. Okay? So, having said all of that, let's go through this pathway, right? And in the end, norepinephrine is much less than cortisol termination. The cortisol now is high here, right? Okay. Initiation starts with the production of pro-inflammatory mediators like leukotriene B4. Leukotriene B4, okay, is a very potent vasoconstricting compound, And it also happens to be extremely pro-inflammatory because of that biophysical event, but also because it leads to the production, ultimately, of pro-inflammatory cytokines in innate immune cells. And then ultimately, of course, in acquired immune cells like T lymphocytes that show up. So you get leukotriene B4 and then prostaglandin 2. Both of those are going to be pro-inflammatory local autocoid or paracrine hormones generated. They'll make it usually past one level of circulation, so they're local. They're happening right at the site of the infection. That increases inflammation until the icosanoid switch is now being shut down. Those are called icosanoids. Icosa just means C20. These are C20 fatty acids. Okay, C20 fatty acids like what? Like arachidonic acid, like icosapentaenoic acid. Arachidonic acid is an omega-6 fatty acid, very common. It comes from linoleic acid in the diet after desaturation and elongation. Whereas icosapentaenoic acid, or EPA, is 20-carbon fatty acid with 5 double bonds of the omega-3 series, where the last double bond is only 3 carbons away from the methyl terminus. The other one was 6 carbons away. And of course, making omega-3 fatty acid, the essential fatty acid there is alpha-linolenic acid. Right? Just so you understand, you can synthesize these from dietary fatty acids. Linoleic for the omega-6 fatty acids, which is the essential omega-6 fatty acid, and the alpha-linolenic, which is the essential 18-colon-3 omega-3 fatty acid, which comes from the diet. Plants synthesize this and sort of lower bacteria and things like that. Okay, but you get a diet from plants, obviously, or from something that has metabolized it to the higher, very long-chain polyunsaturated fatty acids, such as certain marine algae, something I worked on a long time ago. Okay, so the resolution phase is entered. Okay, let's finish it. So at the end of the initiation phase, shown here, there's a lot of LTBB formed. There's a slower prostaglandin being formed. Prostaglandins are made from the what? From the cyclooxygenase pathway. So you have two different oxygenation systems. You have the lipoxygenase, or LOX. There's 3 different isoforms of that enzyme, depending on the position of the double bonds. And then you have the COX, or COX pathway, and you have an inducible one and a constitutive COX. By the way, a good inhibitor of COX, of course, is aspirin, right? Because it acetylates that protein. So it's a suicide inhibitor of cyclooxygenase, just so you remember that. I'm sure all of you do. So anyways, the end of the initiation phase occurs when the levels of PGE2 plus PGD2, not shown here, is equal to molar concentrations of LTB4. So you're getting an alteration of prostaglandin type of icosanoids and the ratio of them to the leukotriene or the lipoxygenase pathway. So this is happening at the local hormone level. The resolution phase is entered, and you can see what's happening is this pro-inflammatory system is changing and the anti-inflammatory system is going up. Pro-inflammatory system dropping during resolution. Okay, cortisol compared to norepinephrine, which is the initial stress hormone, right? It's a catecholamine. Norepinephrine is a stress hormone. Triggers this whole local inflammation response. Same time, you're going through the HPA axis, the ACTH, ultimately down to the pituitary to make cortisol. Now you're regulating the levels of norepinephrine and cortisol. And remember, they're working through receptors, right? Okay. So the resolution phase is entered, triggering the generation of anti-inflammatory mediators, and these include other oxygenated fatty acids known as resolvins and protectins and muresins, and the PGF2 alpha, okay? When the total level of anti-inflammatory mediators, like all those oxygenated fatty acids, because they have a yin and yang to them too. Some of them are pro-inflammatory, like LTB4, and some of them are anti-inflammatory, like this derivative PGF2 alpha. Okay? And of course, that's a COX product, cyclooxygenase product. Total level of anti-inflammatory mediators exceeds a level of LTB4. You see, that's a really pro-inflammatory local. Stop signal takes place. This is the last phase. The inflammation will be terminated by clearing the affected area. That's what you get here. That's your stop signal. So the stress hormones produced by the systemic stress axis, the HPA, right, have a direct effect on the inflammatory phase. Remember, that's the norepinephrine part of it. A microbial invasion, lesion, or injury like a contusion, or even a high stress like a psychological stress, right? All of this can be combined together in something like a car accident or a gunshot wound, right? All those things are happening simultaneously in terms of stress, physical and psychological. Produces then norepinephrine, right? That triggers norepinephrine, which tunes the system to insulin and cortisol resistance. So you don't want the immune system to be blocked. You want it to be turned on. The switch is on, right? So it suppresses cortisol activity. Insulin also has an immune compromising effect. Right? That is, it lowers the immune response at the bioenergetic level, binding its axis of function, which of course is the IR, the insulin receptor. And then all the genes are ultimately regulated there in that given tissue, right? Because that's mobilizing a bioenergetic phenomenon. And that also has to be realigned during the stress. Because during the stress, you don't want to be anabolic. And insulin is very anabolic. Remember, that was the left side of that pathway, right? So, right. So the stress hormones produced by systemic stress acts have a direct effect on the inflammation response, microbial invasion, we went through all of that. The icosanoid switch to resolution can only take place when norepinephrine, that's the stress, initial stress catecholamine, is equal to the level of cortisol, you see? And that's because you're modulating the receptor downstream processing here, plus insulin, and when cortisol sensitivity is recovered, right? So you have to clear the insulin population of bioactivity, binding to its receptor and downstream processing. You also have to clear the cortisol, whatever's in circulation at any given moment, because there is cortisol in circulation at any given moment. Stop signal requires a very low level of norepinephrine and normalized cortisol sensitivity, so you're back to cortisol binding to the receptor and doing what it does. Termination phases, which what cortisol does is it triggers the production of anti-inflammatory transcription and translation ultimately of anti-inflammatory cytokines, for example. At the same time, it suppresses the expression of pro-inflammatory cytokines, growth factors, and transcription factors. Termination phase is entered when the stress axes are all switched off. So that came all from this one paper. Okay. Some papers like it. Here's a paper more recent, Cell Death and Discovery. In about 18 years ago, the SARS, severe respiratory illness, we'll just go through this really quick. I'm going to read it to you. Affecting more than 8,000. It's very limited. It was a very self-limiting virus, with a mortality proportion actually of about 10%. Subsequent identification of a large pool of coronaviruses circulating in bats and other animals portended the appearance of other highly pathogenic coronaviruses. And in 2012, you had the MERS, which is the Middle East Respiratory Syndrome. That caused a similar outbreak with a higher mortality rate. Severe SARS infection manifests clinically as acute lung injury associated with high initial viral titers. Remember that level of virus that we're talking about in terms of like controlling the immune response, right? We want to keep the viral titer down, replication of the virus down initially. Macrophage, neutrophils, or innate immune responses, accumulation in the lungs, elevated pro-inflammatory cytokines, interleukin 1, 18, 6, 8, the MCP1. These are all very powerful pro-inflammatory agents. Recent advances have implicated the inflammatory monocyte macrophages, so the IMMs. We talked about this way back in January, February when I was doing a lot of coronavirus lectures. Go back and listen to those. In the lungs as critical mediators of the pathology. So a superabundance of IMMs is part of the morbidity and mortality of this of the MERS virus, and before that the SARS. Okay, and presumably also this current one. All right. Promotes high initial virus titers and aberrant IMM recruitment. Right. So that's part of the pathology. High initial virus titers and aberrant IMM recruitment. Host disease is likely a combination, directly from this paper, Cell Death Discovery, just published a year ago, not even, before this last so-called pandemic. Get back to this. Host disease is likely a combination of direct viral damage. Now this has, so these viruses do have some very potent virulence factors, and a consequence of an aberrant immune response, which is subsequent sequelae, promoted by these IMMs, right? These very powerful inflammatory monocyte macrophage lineages of innate immune cells, underscoring the importance of determining the mechanisms by which the virus targets innate immunity. So that's what you get from that paper. Here's a paper published now, going back, cell host micro back in 2016. So these are all linear, even though they're published at different times. And they're linear in the understanding. That's why I'm putting them in the sequence of giving you the categorical logic and the dialectic necessary to understand this system, again, at the level of dialectics and ultimately at the level of die-event ontological perspective, which is my way of looking at how diseases, biomedicine work in general. See previous discussions in my lectures. So this journal paper says dysregulated type 1 interferon and inflammatory, here they are, monocyte macrophage responses, the IMS, cause lethal pneumonia in the SARS-infected mice. This is a mouse study, okay? Murine study. Respiratory coronaviruses cause acute lethal disease associated with inflammatory response and lung damage, okay? Robust viral replication accompanied by a delayed pro-inflammatory cytokine type 1 interferon signaling orchestrates inflammatory responses and lung immunopathology with diminished survival in that mouse model. Okay, so that was well known. Now, continuing, interferon 1, this pro-inflammatory cytokine system, which actually basically has a transcription factor now, remains detectable until after virus titers peak. But early interferon 1 administration ameliorates all that immunopathology, the pathology caused by the immune response, like the IMM response. See? This delayed interferon 1 signaling promotes the accumulation of pathogenic inflammatory monocytes macrophages. A delayed interferon response, bad. An early precocious interferon response, good in terms of controlling the disease, lowering viral titer, and preventing excessive morbidity and mortality. Right? That's a take-home message. It results in elevated lung cytokine chemokine levels, okay, when it's delayed, vascular leakage, and impaired virus-specific T-cell response. But we talked a lot about this before. Vascular damage, less oxygenation of the blood, less delivery of oxygen to the periphery, therefore what? Major organ failure ultimately can cause death. There you go. Genetic ablation of the interferon alpha-beta receptor, which is one of the IFNARs, or IMM depletion, which they can do in these mouse studies, protects mice from lethal infection without affecting the actual viral load. How about that? They've separated the viral load, the initial viral load, from disease severity, viral replication, and ultimately the morbidity and mortality caused by the hyperimmune response. They did this way back in 2016. Okay. Overall, this is of course a mouse model, but this is, frank scientific literature. Overall, the murine model demonstrated that interferon type 1, that's the response we're looking at, and the IMM, right, the inflammatory monocyte macrophage lineages that show up at the lung, inducing the hyperimmune response if they're late, promote lethal SARS coronavirus infection and identify interferon 1 and IMMs as potential therapeutic targets in patients infected with the pathogenic coronavirus or perhaps other respiratory viruses. They published this over 4 years ago. And you had to see that after what I was just talking about all the glucocorticoids, and remember in the prenatal pregnant women, remember, and is it okay to use glucocorticoids during this pandemic? And we were talking about how they're using them almost as an initial framework to control inflammation, because women who are pregnant, especially the later stages of pregnancy, right, the last trimester, they have a lot of immune problems anyways, right? In fact, you can get gestational diabetes, which is an immune state. So here, I showed you this many times way back in January. I was looking. We first talked about coronavirus way before it became pandemic. And I was talking about that very paper we just looked at. But early interferon class 1 signaling, right? The SARS disease, you're following it here. You get reduced viral titer. You get a regulated inflammatory response. You get a mild clinical disease. The delayed, again, going down, this is a temporal axis, a delayed interferon response, you get a higher increase of overall viral replication. Then you get the delayed interferon response. You get a dysregulated, because now the IMMs show up, say at the lung tissue bed. You get a delayed IMM response, real pro-inflammatory, real tissue damaging, real destruction of what the lungs are supposed to do in terms of oxygenating the blood and releasing carbon dioxide, right? So respiratory failure, respiratory distress, like RDS, like ARDS, that's what we're talking about here. Lung immunopathology, a lot of cells being killed by the immune system. Now you're even introducing lymphocytes into the process, like natural killer cells and killer T lymphocytes. Lung immunopathology, lethal pneumonia, bad news. No interferon signaling, complete suppression. You get a lot of SARS, but you get no interferon response, and you get a mild clinical disease. Now, how about that? This is a mouse model, but it shows you if the interferon response isn't kicked up at all, if there isn't any pre-existing stress, healthy individuals, for example, with no comorbidities, they can have a lot of viral load, but it goes away because the immune system ultimately generates an immunoglobulin. You start destroying the virus first with the T cell response, after the innate immune response does its functions. So see, that is a very important, that one slide there is very important, I think, clinically. Now this paper, published 10 years ago, okay, Molecular Cellular Biology, has only been cited 37 times in the entire POMC archive. Only cited 37, that means 37 different papers have cited this. A lot more people may have read it, but only 37 thoughts fit to cite it. What does this paper say? Type 1 interferon signaling pathway is a target for glucocorticoid inhibition. What did I just tell you from that 2016 paper about interferon 1, right? It's a target for glucocorticoid inhibition. And there's I gave you the full author names and that paper that you can go back and you can find that paper. Now, what does it say? This is logical progression using dialectics and understanding event ontology, right? Things happening through space and time, right? And happening in real time, event ontological systems. Glucocorticoids are anti-immune response drugs working primarily to control gene expression. We know that that's how they function. The ligand-bound glucocorticoid receptor directly activates transcription of the anti-inflammatory I-kappa B alpha, annexin A1, interleukin 10, and the DUSP1 phosphatase. Remember the phosphatases, which dephosphorylates and inactivates the MAP kinase P38 and Jun N-terminal protein kinase, also known as the JNK. Okay, so that's a real anti-inflammatory paradigm set up here, right, by glucocorticoids. They're all essential to innate and acquired immune responses, all the systems I just told you there. Okay, so glucocorticoids have also been shown to inhibit activating phosphorylation of a protein called TBK. K always stands for kinase, a kinase required for interferon response factor 3 after TLR, toll-like receptor 3 for ignition, right, stimulation. The GR, that is glucocorticoid receptor, interacts with canonical T cell receptors. Now you're talking about the acquired immune response coming in, all from glucocorticoid. We often think only about the innate immune response, what I've been talking about the last half hour almost. But now the acquired immune response, oh yes, the GR interacts with canonical T cell receptors, inducing immunosuppression of the T lymphocyte mediated cascade. So, glucocorticoid receptor directly represses pro-inflammatory cytokine gene transcription through tethering to all these other transcription factors, NF-kappa B, AP1, CREB, T-BET, and NFAT. We talked about all of these in the past, multiple times, way back around Christmas time when I was giving you that whole rather wonderful, I think, series of lectures on the innate immunity diseases. Please go back and listen to those because those all dovetail perfectly into this whole thing we've been talking about for the last now, 5 months, which is the burden of disease associated with comorbidities in the immune response. I talked about lipids. Glucocorticoids are lipids as well, so I haven't left the subject to C. All right. So, GCs, glucocorticoids, tank the transcription of pro-inflammatory cytokines and chemokines, growth factors, and transcription factors. Right? That's clear. Indeed, type 1 interferon JAK-STAT signaling pathway itself is directly controlled. That's when you see type 1 interferon, IFNAR, binding to its receptor and doing the JAK-STAT thing, that is T lymphocytes. Right? So the T lymphocytes, remember, those are the major players for the acquired immune response. Long-term control for immunity because they trigger the B cell response, turn to plasma cells, etc. So that you get high circulating IgG. That's how you become immune, in quotation marks, to further infection. Okay, so I want to finish with this. I want to give you a little bit of just using the dialectic discussions here and a little bit about event logic. Okay, hypostasis is a foundational ontic that is in real time and space event. That's what that word means. Ousia is it means a non-integrated system event. When I say integrated, that means it can't be disintegrated. Okay, it can't be blown apart because it's not composed of parts. It's itself one indissoluble, non-disintegratable unity. But you can have hypostases, which are basically different frames or different persons of one ousia, right? So there are different relations or different ways of looking at it in terms of parallax, if you like the light analogies I use. The parallax, looking at things differently because of light exposure on one ousia, which is the non-integrated, non-dissolvable, right, non-disintegratable system event. So we have 3 hypotheses so far that, and this is very classic. You have hyperimmunity, which we've talked a lot about, which can definitely accelerate disease, morbidity, mortality. Hypoimmunity, which is also really bad because they give precocious infections, and no immunity at the wrong time means you get overproduction of, say, bacterial cells, which outright will kill host cells, or enough of a viral load, which will completely alter the homeostasis of the system, and there's rare events when viruses kill. Or what I call para-immunity, which means that the regulation itself is being modified by an external source. These are terms I've come up with, of course, but I think I like them because they fit together this paradigmatic understanding of a dialectic logical consideration of disease-causing agency. So in one ousia, okay, so you have 3 hypostases: hyperimmunity, hypoimmunity, paraimmunity, because of the indulgence of pharmaceutical grade glucocorticoid introduction to suppress the immune response. And if it's done at the wrong time, it can destroy, for example, that interferon response. And directly, we were looking at the coronavirus paradigm. What you end up with that in the one ousia is the one ousia becomes, remember, it itself exists without parts, is an immunoantinomy, which means it goes against the law. Antinomous, against the law, Latin. And so an immunoantinomy means that the whole system breaks down, right? Because your immune system is now compromised because it is an antinomy to the system. It does not function any longer when you have a corruption of those hypostases. So I'm going to stop there, and let's shut this down here. while I'm figuring this out, um, sorry about this long delay here. What in the heck happened to my slides? Well, we can do this. Did it again. This is going to require my glasses. Sorry for this delay, but this is what happens when you don't have a director. Ensha. Okay, sorry about all of that. I'm not going to be able to cut this out because editing takes a really long time, and I don't think it's that bad for what we're doing, the purposes of this. So I'm going to leave it. Anyways, this is Dr. Dan Guerra. Hoping that you had a stimulating discussion, and bye for now.